Okay, ladies and gentlemen, so here we go with today's biology lesson. I want to apologize for the late post. There have been a series of technical issues. Hopefully, this last time is the charm. And today, we are going to reevaluate or go over some of the concepts that were discussed today in class regarding cell organelles. Now, cell organelles are in the cytoplasm and they're kind of like a conveyor belt. So, the last thing that we talked about was the endoplasmic reticulum or the ER, and the endoplasmic reticulum basically is where the syntheses of proteins and lipids occur. So, real quick to review what the endoplasmic reticulum was, I'm going to type it up here. Okay, endoplasmic reticulum, plasmic reticulum, or ER. Basically, the ER is an interconnected network of membranes. Okay, so we can say is an interconnected network of thin membranes. Is an interconnected network of thin membranes. And basically, there is these membranes form a maze called a lumen. Okay, so in a nutshell, let me actually read from the book. It says the ER membranes form a maze of enclosed spaces. So these membranes form a maze of enclosed um I'm sorry, form a maze called the lumen. Okay, and it is in the lumen and the surface of the ER. It is in the lumen and the surface of the ER where the proteins and lipids are formed. Okay, so that's in the endoplasmic reticulum. Now, today we're going to talk a little bit about the ribosome, right, and other cell organelle structures. But real quick, guys, so we have the endoplasmic reticulum, which is basically an interconnected network of thin membranes. It says that these membranes form maze called the lumen, or enclosed spaces, and a maze of enclosed spaces called the lumen. It says that it is in the lumen and I realized I didn't write the T here, and the surface of the ER where proteins and lipids are formed. So today we're going to talk about another cell structure called the ribosomes. So the ribosomes, which are also on page 72, are pretty much tiny organelles that link amino acids together. So ribosomes are oh no. Okay. Ribosomes. Sorry, I thought I made a mistake there. Ribosomes are tiny organelles that link amino acids together to form proteins. The second thing that is worth noting about the ribosomes is that ribosomes are the site of protein syntheses. Are the site of protein syntheses and active are the both the site of protein syntheses. I'm sorry, ribosomes are both the site of protein syntheses and are both the site of protein syntheses and are active in the process, are active participants in the process. And what this means is that the ribosomes or ribosomes are not just the place where the are not only just the place where the protein syntheses are formed, but they're also active in the protein syntheses. And more specifically, we're going to talk about the rough ER and the smooth ER, which has to do with ribosomes or ribosomes themselves. For example, if we talk about the surfaces of the ER or endoplasmic reticulum, that are covered with ribosomes are called the rough endoplasmic reticulum because when viewed with an because they look bumpy because they look bumpy when viewed under an electron microscope. Okay. And let's see if I can move this over here. Okay. The electron microscope. And what we have after that is the smooth endoplasmic reticulum, or smooth ER. And the smooth ER, they do not contain ribosomes and are not covered by ribosomes and is the site where lipids are formed. So real quick, to recap what ribosomes are before we move on to our other structures, which is the Golgi apparatus. Basically, the ribosomes are tiny organelles, and I can't believe that I misspelled that here, are tiny organelles that link amino acids together to form proteins. They are both the site of protein syntheses and are active participants in the process. It says that surfaces of the ER that are covered with ribosomes are called the rough endoplasmic reticulum because they look bumpy when viewed under an electron microscope. And the smooth endoplasmic reticulum, or smooth ER, are not covered by ribosomes and is the site where lipids are formed. And of course, not just lipids. but a variety of other functions such as breaking down drugs and alcohol. All right. So let's talk about the Golgi apparatus right here. The Golgi apparatus. And let's go straight to the point. The Golgi apparatus. The Golgi apparatus basically process, sort, and deliver proteins. And deliver proteins. Okay. Basically, the Golgi apparatus is the site where processes I'm sorry. Processes sort and deliver proteins. Okay, so that's basically where proteins are packaged and processed. And of course they basically it's where additional changes to the proteins are made. So let's look at the next one. Let's talk about the vesicles. Vesicles, real quick. So we talked about the Golgi apparatus, and it's basically the area, or what the Golgi apparatus does is it processes, it sorts, and it delivers proteins. Now let's talk real quick about vesicles and what they do. And basically, even before I get into the definition, vesicles is just kind of like the way, think of them as storage bags in which proteins are shipped or moved around. But basically, what vesicles do, vesicles divide some material from the rest of the cytoplasm. So they divide some material from the rest of the cytoplasm. Divide some materials from the rest of the cytoplasm and transport these materials from place to place within the cell. And transport these materials within place to play, or from place to place, I'm sorry. From place to place within a cell. Okay, so basically, vesicles divide materials from the rest of the cytoplasm and transport these materials from place to place within the cell. So, pretty much, vesicles is the way that most materials are transported from one protein to another. Now, let's talk about the mitochondria. We're going to talk about the mitochondria and we're going to talk about vacuoles. So, I'm going to go ahead and erase this part. You guys can obviously pause the video and rewind it if you guys have any questions. Now let's go real quick into the other topic, or the other organelles that we're going to talk about real quick. The next organelle that we're going to talk about, like I said, is the mitochondria. Mitochondria. Sometimes the mitochondria is referred to as the powerhouse of the cell. Second thing is that it supplies energy to the cell. So the mitochondria is really important. And when we talk about cell energy in the next chapter, you'll see. So supply energy to the cell. I'm sorry, 3. Supply energy to the cell. And this says they're bean shaped and have 2 membranes. So the mitochondria are bean shaped and have two membranes. And have two membranes. Have two membranes. It says that within these membranes, or I'm sorry, it says the inner membrane has folds. The inner membrane has many folds that greatly increase its surface area. It says within these inner membranes, folds, and compartment, a series of chemical reactions. So this is very important. A series of chemical reactions occur in the mitochondria occur in the mitochondria which converts molecules from the food you eat into which converts molecules in the from the foods you eat into usable energy. So basically, the energy that we get from the different foods that we eat, obviously, because we don't produce our own food, the energy that we get from those foods is actually a series of chemical reactions that occur in the mitochondria. Okay. And now we're going to talk about the vacuoles real quick. Okay, so mitochondria, recapping, summarizing, says is referred to as the powerhouse of the cell, supplies energy to the cell, is a bean shaped. Is bean shaped and has two membranes, and inside of these membranes, a series of chemical reactions occur, which converts molecules from the foods that we eat into usable energy. So I'm actually going to go ahead and erase this again. If you always have any questions, you can pause and rewind the video. Okay, and now we're going to talk about the vacuole. And what is a vacuole? So a vacuole, let me write that down. One is a fluid filled sac. Okay, it is a fluid filled sac used for the storage of materials needed by the cell. Used for the storage of materials needed by a cell. Okay, so is a fluid sac used for the storage of materials needed by the cell. So basically, what the cell needs is stored inside the vacuole. Now, here's some important things about the vacuoles. That vacuoles, or most animal cells, contain small vacuoles. Small vacuoles contain small vacuoles. Contain small vacuoles. And it's also good to mention that plant cells have a central vacuole, which varies different than in animal cells because in animal cells they're smaller. Plant cells have a central vacuole Plant cells have a central vacuole. Plant cells have a central vacuole. Now, what is this central vacuole used for? So, first of all, the central vacuole is a single large vacuole. Single large vacuole that contains, or excuse me, that occupies most of the space inside of the plant cell. Okay, most of the space inside of the plant cell. Let's see if I can move this a little bit so that there you go. Most of the space inside of a plant cell. Okay, most of the space inside of a plant cell. And 5, and the function of the central vacuole is that it is filled with a watery fluid that strengthens the cell. Okay, is filled with a watery fluid that strengthens the cell. That strengthens the cell. That strengthens the cell and helps to support the entire plant. And helps to support the entire plant. Okay, so real quick, 1, the vacuole is a fluid-filled sac used for storage of materials needed by a cell. Secondly, most animal cells contain small vacuoles. 3, plant cells have a central vacuole. 4, the central vacuole is a single large vacuole that occupies most of the space inside of a plant cell and is filled with a watery fluid that strengthens the cell and helps to support the entire plant. Okay, so they're very important in plant cells, to be exact. Although they're also important in animal cell because they have the same function, which is pretty much to store materials inside of the cell, materials that might be needed. So now the next thing we're going to talk about is the lysosome. So lysosomes are membrane bound organelles that contain enzymes and defend a cell from invading bacteria and viruses. Okay, so lysosomes are membrane-bound organelles that contain enzymes and defend a cell from invading bacteria and viruses. Okay, and lysosomal enzymes. So lysosomal enzymes are made in the rough endoplasmic reticulum. The rough endoplasmic reticulum. And that's pretty much it for the lysosome. Now let's talk real quick about the centriole and the centromere. Okay. Now let me go ahead and erase this real quick. What is the centrosome and centriole? Centrosome and centriole. First, let's talk about the centrosome. Okay, the centrosome is a small region of the cytoplasm that produces microtubules in animal cells. Okay, so centrosome is a small region of the cytoplasm small region in the cytoplasm, a small region in the cytoplasm that produces microtubules. That produces microtubules. Two is that in animal cells, it contains two small structures called centrioles. Two small structure called centrioles. Called centrioles. In animal cells, it contains two small structures called centrioles. Now, the centrioles are cylinder-shaped organelles. So you see how we're going to jump from one to the other. Centrioles are cylinder-shaped organelles made of short microtubules arranged in a circle. Cylinder-shaped microtubules um, arranged in a circle. In a circle. Arranged in a circle. In your book, it says that the two centrioles are perpendicular to each other. It says, before an animal cell divides, the centrosome, including the centrioles, doubles and two new centrosomes move to opposite ends of the cell. Now, here's something that I think is worth noting. It says microtubules grow from each centrosome, forming spindle fibers. Microtubules grow from microtubules grow from each centrosome forming spindle fibers. And these fibers attach to these fibers attach to the DNA and appear to help divide, attach to DNA. So apparently the centrioles and centrosomes are really important in cell division, to be exact. And when we start talking about that a little bit later on, we're gonna see how it works in some of these how in more detail. I'm sorry, as to how it works. It says these fibers attach to DNA and appear to help divide it between two cells. Between the two cells, help divide it between the two cells. So basically, what it's saying is that the fibers attached to the DNA. So it says microtubules grow from each centrosome, forming spindle fibers, and then these spindle fibers appear to actually help divide the DNA between two cells. So it actually helps divide the amount of DNA in each of the two cells that are being formed. So again, centrosomes and centrioles are very, very important in cell division. And lastly, and I'm actually going to write that here. Um, organize the centrioles. I'm sorry. Yes, centrioles help organize. Oh, excuse me. Also organize um, microtubules to form cilia, to form cilia and the flagella. Now, what the cilia and the flagella do is they're kind of like the little hairs and the little tail that helps cells move from one area to the other. Okay, so now we're going to talk about plant cells specifically. We're going to talk about two very important structures within the plant cell. So before we do that, let's summarize a little bit what the centrosomes and centrioles do. So the centrosome is a small region in the cytoplasm, or centrosome is a small region in the cytoplasm, and it produces microtubules. It says in animal cells, it contains two small structures called centrioles. Centrioles are cylinder-shaped microtubules arranged in a circle. Four, the microtubules grow from each centrosome, forming spindle fibers. These spindle fibers attach to the DNA and appear to help divide it between the two cells. And lastly, is that the centrioles also organize microtubules to form the cilia and the flagella, which are important in cell movement. So let's go ahead and erase this. And then let's talk about the plant cell. More specifically, we're going to talk about the cell wall and the chloroplast. And cell walls and chloroplasts are two very important structures in the plant cells. So the cell wall um, is a rigid layer is a rigid layer is a rigid layer that gives protection, support, and shape to the cell. It gives protection, support, and shape to the cell. So cell walls are rigid layer. They're not present in animal cells, only in plant cells. And is a rigid layer that gives protection, support, and shape to the cell. Okay, so that's very important. And also is that cell walls of plants and algae have openings or channels. In plants and algae, have, and plants and algae have openings or channels. Have openings or channels where water and other molecules, where where water and other molecules that are small enough other molecules that are small enough can freely pass through the cell wall. That are small enough can freely pass can freely pass through cell wall. Can freely pass through the cell wall. Now, the last structure that we're going to talk about is the chloroplasts, which of course are also found in the plant cells. And the chloroplasts are organelles. So chloroplasts are organelles that carry out photosynthesis. Photosynthesis which basically converts light energy into chemical energy. So chloroplasts are really important because they actually help the cell undergo photosynthesis. Now, your book says basically says like like mitochondria, chloroplasts are highly compartmentalized. They have both an outer membrane and an inner membrane. And it says that they also have stacks of disc-shaped sacs within the inner membrane. These sacs, called the thylakoids, contain chlorophyll, a light-absorbing pigment that gives plants their green color and plays a role in photosynthesis. So we're going to go talk about this a little bit later on. But basically, inside the chloroplasts, we have the disc shaped called the thylakoids, which kind of give plants their green color and also are important in the absorption of light to undergo photosynthesis. So guys, this is the second part of section 3.2. Hopefully this video is helpful and we'll see each other in our next class. So stay safe and we'll see each other soon. Bye-bye.